Good afternoon. My name is Adriana Link, and I'm the head of scholarly programs at the American Philosophical Society. Welcome to today's virtual discussion with Neeraja Sankaran on her new book, A Tale of Two Viruses, Parallels in the Research Trajectories of Tumor and Bacterial Viruses, now out from University of Pittsburgh Press. I'm glad that so many of you have joined us today. The American Philosophical Society resides in Lenape-Hoking, the homeland of the Lenape people, whose historical relationships with the land continue to this day. The APS acknowledges with respect their continued presence and perseverance and expresses its sincere thanks for the past and ongoing generosity of numerous Indigenous communities and individuals who have offered their guidance, expertise, and opportunities for collaboration. For those joining us for the first time, the American Philosophical Society was founded by Benjamin Franklin in 1743. The Society is a catalyst for the discovery of new knowledge. Election to membership honors those who have made exceptionally significant contributions to science, the arts and humanities, and public life. The Society promotes research by providing over $1.5 million in research grants a year, primarily to younger scholars who need the support the most. Our library and museum, collections, and research centers serve scholars and visitors from across the globe. Please check our website, www.amphilsoc.org, to learn more about what we do and for news of forthcoming events. We're using Zoom webinar for today's discussion, so not to worry, you've all been muted. If you have a question for today's speaker, and I hope you will have many questions, we encourage you to use the Q&A button at the bottom of your navigation bar. And you can type your question in at any time during today's discussion. There will be time at the end of the panel for questions with our speaker. We're also excited to offer closed captioning for today's virtual discussion. If you would like to use it during the panel, please click on the CC box on the bottom navigation bar of Zoom. It is to the right of the Q&A button. With that, I'm very pleased to introduce today's speaker, Neeraja Sankaran. Dr. Sankaran is a historian of science and medicine with a primary focus in the recent and near contemporary history of biomedical sciences. She holds a PhD in history of science from Yale University, a certificate in science writing from the University of California, Santa Cruz, and a Master of Science in Microbiology from the University of Alberta. Her recent books include Leeuwenhoek's Legatees and Virology's Beneficiaries, as well as the book, which is the topic of today's discussion, A Tale of Two Viruses. An independent scholar, she has held positions at universities throughout the world, including the United States, and I should say that she was a fellow at the American Philosophical Society, as well as Egypt, South Korea, India, the United Kingdom, and the Netherlands, where she is currently a research scholar at Utrecht University. And with that, I will hand it over to you, Neeraja.
Hello, and thank you all for coming. This is a great pleasure to be here. As Adriana mentioned, I have been a fellow at APS, specifically to do with this book, which is why it's so exciting to speak about this book to this audience. I'm going to pretty soon share my screen with you and plunge into the book right away. There it is. Please bear with me for a second. And all right, so my book, and I put the picture of the APS, the facade, which I pulled from your website, on my cover, because what I'm talking about, see, I've written this book, I've spent the better part of the last decade thinking about writing, doing research about this book. And so I can talk about it for as many hours. And since I only have 20 minutes to actually talk about it myself before getting into the more interesting part, which is to have a conversation about it, I figured I'd focus in on those parts of the book for which I relied heavily on the archives at the APS. So, and I'll talk a little bit about where my title comes from, and also how, it's something that was brought up in the blurb for introducing the book. What do Dickens and the APS have in common? Well, you'll see in this book, A Tale of Two Viruses, very not so subtly drawing on Dickens's very famous title, A Tale of Two Cities. So which two viruses is the first obvious question. And as Adriana already mentioned, the two viruses, or the two groups of viruses, I should say more accurately, are the tumor-inducing viruses and bacterial viruses, better known as the bacteriophages. And the reason I figured that these two viruses deserved a tale to be told about them is that the trajectories they shared, the research trajectories that they shared for a large part of the 20th century, from the early decades on until about the 60s, was very, very parallel. There were lots of parallels. And then sometime in the late 50s through the 1970s, these parallels converged, and then the two viruses went on to become famous in their own ways. The bacterial viruses much earlier, actually, bacteriophages, but the story ends then. So I'm going to start with giving you a couple of slides that show the story in brief. And these slides are actually, I would say, equivalent to what I presented a long, long time ago. I think it was 2011 at the HSS, History of Science Society meeting, that I basically made an entire talk out of what I'm showing in this, literally the first slide. And basically the idea is that in 1911 and in 1917, there were two discoveries of a transmissible tumor, a sarcoma tumor, that is a tumor of the connective tissue in chickens, and this, again, a transmissible lysis, that is destruction of bacteria. These discoveries were made completely independently of one another in completely different research contexts. One was almost a pure research context at a research institute. The other was made in the context of a garrison, of an outbreak of a disease, of a dysentery, small epidemic, if you would, or a small outbreak outside of Paris. Both people who investigated these phenomenon thought that the causative agent was a virus, and the reason I say virus in quotes, whatever a virus meant then. And the reception, by and large, in their respective communities was a resounding no. It's not a virus. And the reasons that the people gave were astoundingly similar to one another in many ways. And then eventually the parallels started to converge with the resolution of a phenomenon called lysogeny, which I've marked out here, 1953, which for many years, it's an amazing paradox actually, because without going into detail, lysogeny was, since 1920, the main reason touted against the reason why bacteriophages could be viruses by the person who discovered this phenomenon. And I will go into detail if you have questions about it. And then the ideas embedded in lysogeny were carried over the next almost 20 years to the case of the cancer viruses. That came to a head finally in 1970. And that's the point at which this story, My Tale of Two Viruses, ends. So, like I said, this is the story in brief, covered through 7 chapters in my book. Now I'm going to go to the beginning of the Tale of Two Cities. I pulled this down, and if it's a little difficult to read, everybody knows most of it anyway. The opening paragraph, it starts with probably the most famous opening of all times. It was the best of times. It was the worst of times. It was the age of wisdom. It was the age of foolishness. It was the epoch of belief. It was the epoch of incredulity. It goes on and on. It was the season of light, the season of darkness, the spring of hope, and the winter of despair. Well, back in 2011, I think it was, when I first presented it, I kind of threw this out. I didn't call my talk A Tale of Two Viruses, But I threw out the idea there very tentatively and as a throwaway line, and just stopped with it was the best of times, it was the worst of times, because that's all I'd remembered. And a friend of mine who is unable to attend today, probably because she's in an airplane, said to me after the talk, you need to take ownership of that. There's no need to be so diffident. And I took her advice to heart. And the first thing I did was, when I got back home, look up A Tale of Two Cities. I happened to have it on my Kindle, or I bought it on my Kindle, or got it for free off the Gutenberg Project. I'm not sure. And I was, to my delight, when I read this whole paragraph, the third line, it was the epoch of belief, it was the epoch of incredulity, made me come to a full stop. I'm like, hey, that's exactly what was happening with the guys I'm telling the story about. So here was a question of life imitating art in a very unexpected way. So of course, then it became the trope, the major literary trope for my book. And I used it. And as you can see, I've quoted it in my preface with until the epoch of incredulity. I wanted to stop there. And I've actually used that phrase again in naming a chapter, and other phrases from the book in naming different chapters. And then there is one other cameo appearance by one of its characters in the very last chapter, but I'll talk about that when I get to the last chapter. So like I said, best of times, some of the best times working on the book were inside this library, which is why I brought up this slide in a kind of cheesy homage to the Institute. But here are the specific APS connections, the archives, the people whose archives that I relied most on at the APS were Peyton Rous, James B. Murphy, Thomas Rivers, and Thomas Anderson. And the reason there's a picture of another institute down below is that all these scientists actually worked at the Rockefeller University, and these are the gates to that university, and Upper East, the lower Upper East, so to speak, 66th Street and York Avenue in New York. So those are the specific APS connections, and the reason is that all of the early scientists, as well as board of directors of the Rockefeller University, their papers are archived at the APS. So here are the 7 chapters of my book. And what I'm going to focus on, like I said, are the chapters that that use the APS archives most heavily. And those are chapters 1 and 2, and to a slight extent, chapter 3, and then chapter 5. And this happens to mirror the way I structured the book. And I'll tell you a little bit about that, since this is a parallel history. The first 2 chapters talk about the recall to life, incidentally, is another trope straight out of the first book, as they call it. The first chapter or book in Tale of Two Cities is called Recall to Life. But those were, so the first chapter deals with the discoveries that happened 1911, 1917, within a few years of each other, and how the people conceived of their discoveries. The epochs of incredulity go a little farther for the next decade or so, the immediate aftermath, and how mostly it was incredulity, though there was some belief as well, else I don't know whether the scientists would have gone on. And then in chapter 3, I kind of stop the main narrative and go back and lay the backdrop, because the backdrop against whether or not these things were viruses or not is a question of what was a virus. And this is something, this term has undergone a lot of morphing, to use a modern terminology, variance in meaning, to borrow something that a very renowned historian of science has used over time. When it was first brought into the English language, it actually meant poison, but it started to mean something very specific, evolve into something very specific, really not till chapter 7 of my book. But then after that, I broke the pair. I didn't deal with both viruses in both in the next 2 chapters. I dealt with viruses in turn, in part because at that time there's a temporal break. The parallels happen, but they're stuttered parallels. The bacteriophage is a brand new discovery. It's research in 1917, and the research on it sort of continued and took off. Like my first chapter heading suggests, that tumor viruses were more or less, that idea was recalled to life in 1911 after it had been declared dead. It was declared dead yet again, and then it's reawakened, which is why I call that chapter Reawakenings, in a sideways nod to a movie about a different viral disease called Awakenings, not a book, sorry, and only then a movie. And then again, chapters, so Reawakenings is all about the viral cancer virus portion, tumor virus. Chapter 4 was all about the bacteriophage. Chapter 6 again goes back to it's becoming a second kind of intermezzo and takes this whole notion of the evolution of virus forward. And the reason that's broken up into chapters 3 and 6 is not only chronological, but also because in the second half, which begins in the 1930s, there's a whole explosion of different technologies, which really, and the birth of molecular biology, which really changes how one could start to think about viruses. And ultimately, and in a final homage to Dickens, the final chapter is called Knitting Done. And I will bring up the reasons for that knitting metaphor, time permitting, and how lysogeny served as the linchpin that kind of brought everything together and allowed me to get the knitting done. So that's the basic outline of the book. So here is the called or recalled to life. And the ideas, so the epigraphs, I basically excerpted, snapshotted the title page of each chapter with the epigraphs so that it'll give you. And what Peyton Rous said in his very first paper, so backing up, Peyton Rous was a scientist at Rockefeller University who'd been hired specifically to work on problems of cancer. He was trained, he'd been trained as a pathologist, in medical school, and a pathologist. And he had been explicitly warned not to work on the cancer problem, but was persuaded to join the Rockefeller to do that, even though the Rockefeller was also explicitly not a cancer research institute. It was a biological research institute, a medical, sorry, it was then called the Rockefeller Institute for Medical Research. It is now, sometime in the 60s, it became a university. And it's a very unique institution to this day. And one could write an entire history about that. But Peyton Rous, one day, some worried farmer, this is exactly how the story goes, a worried farmer walked in the gates with a hen, which had this lump protruding from its breast. When I told the story a week ago to Babak Ashrafi across the street at the consortium, and it struck me then that you couldn't do that anymore. Just because somebody is worried about something their hen or their pet would have, they couldn't just walk into a research university saying, hey, what's wrong with this? You could in 1910. And so Rous began to investigate it and found out that it was a very different kind of cancer than anything that had been observed before, because not only could you transmit the cancer by placing bits of the tumor in an unaffected hen, but if you ground up the tumor tissue and filtered it, you could transmit it even with the cell-free filtrate, which is why it led him to think about it, think in terms of a virus, that it was an invisible living thing because it could propagate very clearly in the new thing. So that's why he thought it was a minute parasitic organism. And meanwhile, in 1917, Felix Darrell, who discovered the bacteriophage, also talked about it in terms of viruses. He called it a microbe. This is translated from the original French, but he gives a very similar explanation for his discovery. So here's the picture. I should have probably jumped to this photograph. This is a photograph of Peyton Rous about that time. He's not a very young man even then. And this is the picture, if not of the original hen, then these were Plymouth Rock hens, and the tumor was very specific. It couldn't be caused in other hens, not with the extract from this. But this is the kind of tumor that was caused. It's a connective tissue tumor. Over on the other side of the pond, but slightly south in France, in the Pasteur Institute, this guy, Felix Derel, was able to discover these. So the lawn of bacteria with these clear holes are caused by what he called the bacteriophage and thought was a virus. That is a minute parasitic organism, in his case that he said, lives at the expense of bacteria, which is why he called it a bacteriophage. So the first chapter is all about the discovery and why they thought it was a virus, which I hope I've summed up quickly enough. Chapter 2 moves on to the epochs of incredulity. And here I'm going to focus in on Rous's incredulity, the reception that Rous got, and not too much about Derel, except to make this one quote. His staunchest opponent to the end of his own days was this already very eminent scientist, Jules Bordet, who said the invisible virus of Derel does not exist. James B. Murphy, and I mentioned him in the list of Rockefeller scientists, once wrote to one of his students saying, I never believed in the virus theory. Now, for James Murphy not to believe in the virus theory was actually quite a blow, would have been really difficult for Rous, if one thinks, because Murphy joined specifically to work with Rous in 1912. And so there was a lot of dissent from within. Now, nevertheless, the two men never aired out their differences, at least in public. It only came out later. And throughout their writing about it, they called it an agent. They never used the word virus. And partly this was Murphy's resistance to the idea. Here he is. This is James B. Murphy. And again, but he played a very important role in keeping the chicken tumor problem alive at the Rockefeller, and indeed alive anywhere for a good number of years. And the reason is that Rous by 1915 had grown tired of trying to find an agent. You know, people couldn't visualize viruses back in the early 1900s. And he couldn't find an equivalent in mammals. He couldn't find an equivalent in other animals. And he gave up, and he had another problem that he had to attend to. So he just shifted course in 1915 and never returned to work with the Rous sarcoma ever again, although he did return in a big way to cancer research, which becomes part of my Reawakenings chapter. But soon after Rous had retired from the problem, Murphy was all set to give up as well and move on to other issues of cancer research, or actually abandon cancer research altogether when the Rockefeller was endowed with money specifically for doing cancer research. And by then Rous had moved on, had already started on a new project. And so Murphy was asked whether he would care to continue. And he said yes. This was very soon after Rous had left in 1915. But for many years did not work on the sarcoma virus, in part because he didn't believe it was a virus, but more than that, he was doing other work and went on to, establish a very solid reputation as an immunologist and his work on lymphocytes. He did work on lymphocytes and their role in whatever they did for during the cancer process, but he didn't work on the sarcoma problem particularly until a few more years. And that is the first reawakening. So I'm going to jump to, before I jump, however, to chapter 5, the background chapter, what was a virus? I'm not going to mention too much about it because much of that work took place outside of the Rockefeller. In fact, all over the world, there were discussions about what a virus was. It begins with the discovery of a plant virus already in the 19th century. But one of the prominent players by the 1930s was another Rockefeller scientist called Thomas Rivers, who is the first generation of people to explicitly call themselves virologists. Rous was a pathologist, as was Murphy. They were not virologists. I mean, virology was not yet a discipline. But so that's a lot of that chapter is full of background. It's the backstory. and what forms the context for all discussions. Is this a virus? Is this not a virus? And do people even agree what is a virus? Which is why I called the chapter, What Was a Virus? And as one person says, we do not know what a filtrable virus is. And before that, in 1890, Louis Pasteur said, in summary, all microbes are viruses. So, all viruses are microbes, or whatever he meant by that. But moving on to the reawakenings then, in 1925, this person, and I'm going to show this picture, there's a person, his name is William Guy. So one of the things I should mention, back up very quickly, Rous, though he stopped work on the sarcoma altogether, he maintained a supply. He would he would grow more sarcoma tissue and maintain a culture of the material, in hens, in his lab, and sent it to whoever was interested in working on it. And in fact, there's a large, I think, multiple folders of letters, correspondence with various people who wrote in to request for material as part of the material that I found at the APS. And it could make a story. But one of the very interesting exchanges he had was with this guy, whose name is Guy, G-Y-E, William Guy. And I neglected to put everybody's names on this picture. I apologize for that. But his name is here in the center as well. Guy wrote to Rous for the first time in 1923, requesting for the material. And by 1925 said he had evidence that it was indeed a virus. And that actually revived worldwide interest in this problem of the chicken sarcoma. And Rous would later in life say that his work on sarcoma virus, or the sarcoma virus anyway, was saved by an Englishman. A second person with whom there was a lot of contact was somebody who was Guy's student for a time, Christopher Andrews, who is a virologist, a virologist, who actually had visited Rockefeller prior to that, while still a medical officer, and had worked on other things. At that point, had struck up a friendship with Rous, and in fact, they maintained a correspondence for decades. From the 30s or even late 20s all the way until Rous died, literally. So until the early 60s. Until the late 60s. Guy died a lot earlier, but there was this conversation, and literally throughout this period, through the 20s and 30s, the notion of that cancer could be caused by viruses was kept alive. Rous remained very interested in the work, though he never did any work on it during this whole period. And at one point, he actually wrote this in a letter to Guy. We are the three musketeers, and he includes Andrews in that, and must carry more than blunderbusses, by which he means we need to have real fine arguments. It's not good enough to just believe it. We need to try to keep producing not just evidence, but arguments in favor of the viral theory. His opponent, Murphy, his opponent in idea that it was a virus, who nevertheless carried on work, like I said, the continuity, also had some amount of correspondence, especially with Andrews. It was good-natured, often, but also contentious. And it was certainly never the close friendship that Rous and Andrews developed over time. Guy said a lot of things that weren't necessarily correct, but at least in the early years, Rous certainly relied on him to bounce ideas off. There would be things like, okay, sit back and listen to the story. Letters would literally say that. Andrews had worked on a number of different viruses. In fact, he is one of the first linchpins because he actually worked on bacteriophages as well, and on similar problems like determining size, and worked with another scientist who did a lot for bacteriophages in a chapter that I don't mention here at all, which is chapter 4, which in the book is called Romancing the Phage. You can see I have a sort of fondness for making allusions to popular culture and to literature, but actually was the in many ways the beginning of the book, because chapter 4 is my dissertation all scrunched up, or most of chapter 4 is a large part of my dissertation scrunched up. And so was the inspiration in some ways for what I noted there, and then became this very large book, which has, I mean, Rous and all don't make any appearance in my dissertation except in one footnote. I'm sorry, why is, yeah. So one of the letters that came by, which was one of my big discoveries at, well, I knew it was there. Somebody had already told me, but it was something, I was the first person to do something with it. And within a year of my first visit to the APS, I had published a paper about about this one-page letter, or rather fairy story for oncologists that Andrews wrote to Rous, in which he uses notions, he uses various fairy tale tropes and metaphors to conjecture how viruses could be causing cancer. And for this, the mechanism he borrowed directly from the then existing ideas about lysogeny, which I already said was one of the greatest arguments against a viral theory of bacteriophage. But Burnett had had suggested a compromise without being able to exactly tell you why or how, because those tools weren't available yet. And this Christmas story begins, and I'll just read out the beginning because it's just so lovely. Once upon a time, there was a family of viruses, much like other viruses. And then he talks about them losing their jaws. I believe this is actually a typescript in Rous's files, but the original was obviously handwritten, but Rous then had them typed up. And these are in Rous's own handwriting, where he's written, a sprig of holly was drawn in here. And so I added a little sprig myself, and by C.H. Andrews. And it's a one-page article. It's a one-page fairy story accompanied with a letter full of conjecture. Prior to Rous's own delivery of a very prestigious lecture called the Harvey Lectures in New York with the Harvey Society, which, even though it had been many years since he discovered the what would eventually be called the sarcoma virus, he chose to speak on viral etiology of cancer at the Harvey Lecture, and got a lot of flack for it from some people. Very good friend of Murphy's, but not Murphy himself. So that was one story that I thought I would let. And so this was actually my first stepping stone. The paper I wrote based on this was my stepping stone to writing the book. I didn't start at chapter 1, really. I started by writing this paper, and eventually this paper became part of chapter 5. And then there were letters, like I said, if you backed up a little bit, there were these letters between Andrews and Murphy, also speculating. And Andrews tries to persuade him, look, you're saying that a mutagen. So Murphy's idea was that this cancer-causing agent was a mutagen and not a virus. And Murphy's trying to tell him there's not really any, they're not mutually exclusive. And he writes this rather detailed letter. And in 1933, he actually wrote another letter in which he's suggested to Murphy, he says, I'd suggest you make a New Year resolution to start calling the thing an agent. And Murphy writes back saying, I'm shocked by your suggestion. Don't you realize I'm essentially a kind person and wouldn't think of doing anything to deprive other people of pleasure? And you must know with what zest Tom Rivers includes the chicken tumors among virus diseases, because he thinks he annoys me. So, there's banter back and forth, but Murphy really has no time for this. And in a later letter, shortly thereafter, when he writes something to locally to a friend called Ewing, he says, I don't have time. I've given up arguing with Andrews. Meanwhile, Andrews says something similar to Rous about Murphy. So but there were never personality clashes, there are ideas clashes and a lot of good-natured banter for the most part. But to back up one moment, I should say that the oncologist in what he called his fairy story for oncologists were very clearly, even though he never explicitly says so, are Murphy and this other person, Ewing. And fast forwarding now to the final chapter, lysogeny becomes the linchpin. And the only reason I want to bring this one up is really to go to Dickens, because when I reached this chapter, and I floated this idea to somebody before I included it actually as a book, I found to my utter delight that the bacteriophage, the way it was becoming, and the same way that the bacteriophage was becoming, causing lysogeny, and the way then ultimately the cancer virus is able to induce a cancer, is to earmark its victim by knitting the DNA, its nucleic acid, into the DNA of the host cell. So viruses are viruses because they're not capable of independent living. They're obligate parasites, and so they live. Yeah, and if I'm going over time, please stop me, Adriana. But very quickly, so they knit themselves into the genome. And this is something that Andrews brings up in his fairy story as well. But lo and behold, if people remember Tale of Two Cities, one of my favorite villainesses, literary villainesses, characters is, well, there are two of them. One of them is Madame Defarge, who's in the Tale of Two Cities. She's this horrible woman who attends every guillotine session, sits there and is constantly knitting. And what she's doing is maintaining a list of the guillotine victims on whatever she's knitting. Well, I couldn't have asked for something more apropos for, as you know, that metaphor. So lysogeny, the bacteriophage is then therefore the bacterial Madame Defarge. And a very talented microbiologist, cartoonist friend of mine came up with this cartoon, where if you can see, the bacteriophage is there supplying the wool to become this, supplying the threads that then become part of the bacterial genome. There's the bacteria, and there's a guillotine, just to remind you that this bacteria is also watching guillotine victims. So with this, I shall end my talk and open it up for questions. I hope I haven't gone on too long.
Thanks so much. No, this was great. It's wonderful to see sort of the whole project sketched out in this way. And I encourage those of you who haven't read the book yet to pick up a copy. I think it really does a nice job of laying out the sort of longer history of virus research in the first half of the 20th century. So there's some great questions coming in already, many of which mirror my own. So I think I'll start with one actually from one of our audience members, which asks, says, fascinating talk. I wonder if you could talk a little bit more about why this particular animal-based sarcoma and why it so captured the attention of these important scientists. Also, historically, why do some diseases get scientific study when others might not? So thinking about this question, what are the kind of questions and problems that are driving biomedical research in the early part of the 20th century? And why chickens and tumors? What's going on?
Okay, so why chickens and tumors? The farmer came and brought, it was opportune. Rous was there to do cancer research. He hadn't come up with a project yet. He was very newly employed at Rockefeller when along came this farmer. Nobody else was taking this on. Rockefeller, like I mentioned, was actually a very new institute itself. It was it had just opened up in 1901, and this is 1910, within a decade. And so along came this tumor, and he started investigating and kept on only for 3 years, as it happens. He later turned to a very different tumor. When he returned to the tumor virus problem in the 30s, he started to work on rabbit papillomas, which is the other animal mentioned in the fairy story, I should say. So why are some diseases studied and others not? Well, cancer has always been a disease that has interested people, right? And there's always been an agenda. I mean, there was the war on cancer declared in the mid-20th century and has been carried on ever since. There have been other virus diseases, but incidentally, that cancer could be caused by a virus was an absolute no, not only because people didn't believe it, but also because virus signified infection, and that sort of brought a stigma, and there was no way that that stigma should reach cancer. And also in 1910, just before actually Rous starts to publish material, started to investigate, there had been but I'm digressing. But there had been an announcement, a consensus at an international cancer congress that the parasitic, the notion that any parasitic or bacteria or invisible organism, that any kind of parasite could be causing cancer is absolutely no. It's not an infectious disease. And that was that held sway for a very long time. But to get back to the main question, what problems are chosen and what are not depend on where the money is. People, if they don't get money, they cannot continue to research stuff. Bacteriophages, for example, were very interesting to Burnett in the 20s and 30s, but then he moved on to animal viruses, partly because he himself was interested in medicine, but then he got a lot of money from the Rockefeller Foundation to pursue medically important viruses, and bacteriophages were not yet the tool of molecular biology. That wouldn't happen, and that would have anyway made him leave it. But again, so there, where money comes from, what is having an interest, sometimes it's personal interest, right? You get into doing research for a personal reason and you get the right results. Sometimes there are too many dead ends. I mean, the reason Rous stopped doing cancer research by 1915 is there was a dead end. He could not find what he was looking for, and he thought that the negative results were very discouraging. And he felt, yes, it was worth pursuing, but in the face of this kind of something else needed to give before he could go back to the problem. I'll stop talking and let somebody get to the next.
No, I think that's great, and sort of what that gestures towards is, the title of this book is A Tale of Two Viruses, but in some ways too, this could be read as a tale of two institutions, sort of grappling with this question of what is a virus and what it is not. And sort of on that note, one of the things that I really appreciate in the book is the way that you show how much the role of visualization and efforts of researchers to make viruses something that could be seen and observed and studied played into the identification and acceptance of them as sort of a definable unit or thing or entity. Which also sort of goes back to this theme of incredulity that you raised at the beginning of your remarks. So there's other questions in the Q&A about sort of what processes were being used in the lab. So what were some of the different processes and techniques involved with making viruses visible, and how did those change during the period that you cover in your story?
Okay, so in the beginning, there were viruses were invisible. In fact, in the 30s, Tom Rivers expresses with great frustration that viruses are defined in negative terms. They cannot be seen. They cannot be held back by filters. They cannot be grown. And of course, the virus depends on its property of not being able to be grown. It wasn't until the development of certain techniques, to begin with ultramicroscopy, so to be able to see smaller and smaller things with different kinds of cleverer mechanisms of, ordinary microscopy, but ultramicroscopy with UV, you got to smaller and smaller things. Filtration, ultrafiltration actually is something that is not given any, people talk about filtration, but the notion that a virus was filterable, well, exactly how filterable was it? Bacteria are filterable too, if you use a small enough pore. I mean, it really depends on the size of the sieve, right? You can, when you're sieving tea leaves versus something with lumps, in the kitchen, even if you think about it, your sieve size changes depending on what you're trying to, if you're just trying to wash veggies, you use a very broad colander versus you use a much finer mesh. So, and the bacteria, once you were able to actually know the size, then you could start to try and measure but the viruses eluded even that, but there were bacteria you could measure that way. Eventually, you were able to make spore sizes small enough to hold back some viruses and see what viruses could pass through. And that was more or less the first visualization of viruses. At least you got an idea of, okay, it's smaller than this size because it can pass through that hole, but it's bigger than X because it can't pass through that hole. And then there's the X-ray techniques that come in from X-ray diffraction studies and crystallography. And in the 30s, the first kind of visualization of anything happens with the visualization of molecules. And this actually leads to an at first erroneous, but later then it also clarifies the matter that viruses are made of protein. Because at first you crystallized the shell. Eventually, through X-ray crystallization, you were able to figure out that the virus actually has two parts, an outer something made of protein and an inner something made of nucleic acid. Of course, you also needed to discover the fact that nucleic acid, DNA actually played a role in all these things. That didn't happen till later. That DNA has, knowing a little bit about the structure of DNA, because proteins are so big, it was easy to lose sight of the fact that there was DNA or RNA inside it. So there's that kind of visualization. And then the ultimate visualization, of course, is electron microscopy, which happens in the mid-30s. And electron microscopy did for viruses what Leeuwenhoek, who's in the title of my other book, Leeuwenhoek back in, the time when the Royal Society was first instituted. So 16th century, sometimes I get those centuries wrong, 16th or 17th, in the 1600s, 1700s. What Robert Hooke and Leeuwenhoek did with ordinary light microscopes. And that's how they discovered bacteria. And, so that's when so the light microscope opened up this whole new world. And the electron microscope then did the same for virology 400 years later. So those are the different techniques. And then another very important technique was the ability to grow viruses outside living cells. So the first viruses that one could grow and actually assay and look at, you knew what was happening if they were growing, were bacteriophages. That's how you found them. So these large holes in a smooth lawn of bacteria is one way of visualizing what bacteriophages do. And then they did something similar, caused similar pock lesions in the plant viruses like tobacco mosaic virus and other ones. And it was easy to assay those. But the animal viruses were very, very elusive. And that's partly because one didn't exactly know how to grow animal viruses in the lab. And until you had good, stable systems for growing them, then only you knew how they looked under normal circumstances and if viruses were growing. It was only then that you could actually think about looking at viruses. Before that, there was a period when people were growing viruses, injecting viruses onto the chick embryo membrane and growing, and so where they formed pocks, not unlike the pox as in POCKS, which is not unlike, and they were equivalent to the plaques of the bacteriophages, which were themselves perceived as equivalent to bacterial colonies when you grew isolated bacterial colonies. So there were different techniques for visualizing different levels. It's like visualizing bacteria. You look under the microscope, you see bacteria, but you see bacterial colonies, and those tell you a lot about bacteria, which looking at an individual does not. And, so it points to the importance of population versus individual studies, and all of that's really so there were all these techniques, and a lot of my chapters 3 and especially chapter 6 go into that, all these background items. Specifically, how these viruses were studied. Do we have time for that? I've talked a little bit about bacteriophage, and I mentioned the fact that you studied cancer viruses by their ability to cause cancer. And then eventually, when you were able to assay them on a plate, that's when actually you were actually able to get a lot of information. When you were able to culture them is when you were able to find out other things about them. But it was the search for this virus with an electron microscope, by the way, which was happening in Murphy's lab, that led to a whole new problem and the creation of a new element of cell biology, the discipline of cellular biology at the electron microscopic level, at the substructure level. It was born out of the search for the Rous sarcoma virus. So
maybe a related question, and a good one from one of our attendees. So we've talked a little bit about the sort of experimental process and the role of visualization in coming up with sort of the circulation of new scientific ideas. But you also had that really wonderful slide where you showed the three musketeers and their correspondence. So this attendee asks, what do examples of correspondence reveal about the scientific research process in the 20th century? And how similar or different is that process today in an age of digital communication and data sharing?
Okay, so I rely so heavily on correspondence. I couldn't dream of doing research without that. And it's such a wonderful snapshot into the minds and the language and the world of that has been, right? That is no longer with us. And some of those letters jump out at you as they could have been written yesterday, except that they're handwritten. And I don't know that people anymore. When I was a kid, I still wrote letters by hand to my friend, right? And we actually maintained a correspondence between India and Brazil. So this was when I was I don't think most youngsters today do that. Everything now, certainly I don't anymore. I mean, so I think the main difference is today it's instant. You send an email, which means, I mean, if you were to track emails, I'm sure there would be a lot of really interesting correspondence, but how many people hang on to their emails and are willing to archive them? I don't know. You can edit them though, which you can't do to older letters except strike with a pen. You can clean up your writings, although I notice a lot of bad, spelling errors. And when I'm dictating and talking, those kind of errors, they creep in, and sometimes they're there for posterity, which makes for fun things as well. But definitely correspondence tells you a lot, because depending on whom they're corresponding with and at what level of formality, you also get a great picture of how people are thinking. So this I especially found during not in the correspondence that I quoted here. Here it's all between scientists who are working, but as you can see, a letter, I mean, he shares this fairy story, right? That he says is not for publication. And in fact, when I published the paper, it's the first time it was published. But at that time, he kept it private, and Rous circulated it to select friends. He didn't want to rub any people the wrong way. I assume he didn't want to completely insult Murphy. Although in another letter, he does ask Rous, is my name permanently in the mud with Murphy? So there's a lot of humor and banter and that sort of stuff. But I remember when I was looking at the Burnett, Macfarlane Burnett's letters to his wife, to his then fiancée, he would be writing about bacteriophage nonstop. In fact, one of the letters has and the reason I call that chapter Romancing the Phage is that one of the letters has this beautiful epigraph that says, onto the bacteriophage, your chief rival for my affections at this stage. Has to be a very nerdy 20-something, 30-something writing that to his fiance, who's a nurse or school teacher, certainly not a biological researcher. But besides that, he explains things to her in ways, when he's trying to explain the phenomenon of lysogeny, he sort of uses daily examples of disease. Diphtheria was big then. And he says, well, the bacteriophage is living inside the bacteria the way dip lives in a carrier who doesn't have symptoms of the disease, but is able to pass it on to somebody else. So you're able to test ideas, use informal language, use analogies, think about using analogies. Now, Burnett, for that matter, Andrews, who, by the way, worked in labs next door to each other for a while, which is how this bacteriophage idea gets carried over to the cancer idea and becomes the fairy tale, in my opinion. At least that's the chain of thought. But so letters reveal a lot of background information, right? I mean, a lot of what Rous, Guy, and Andrews said to each other never makes it onto a paper, but they're really testing ideas and honing arguments. So I think, it gives us a lot of interleaving. What Lawrence Durrell writer of some very fine fiction called the interlinear. So you have a narrative and then you fill it in with other sources, with information about the same period from another perspective. So you provide these interlinears. And I think without those kind of interlinears, history would be very boring. The doing of history, This
is wonderful. We're very quickly running out of time. So I'm going to try to put together two questions that I want to make sure that we touch upon. And one of them echoes a question from an attendee. So it's very clear that APS's collections played a very major role in the writing of this manuscript. And you and I have often talked about the sort of incredible set of resources at APS for folks who are researching and teaching topics in the history of biology and medicine. So part one. Part two, viruses are very much in our daily discourse in the time of COVID-19. So thinking about what kind of lessons or teaching opportunities exist in telling the story of viruses, how might you frame a course on the history of biology using APS materials? And then more generally, what does your story have to tell us about the way that we might grapple with the implications of viruses like COVID-19?
Okay, I can answer the second question first, if I may, only because I can do that rather quickly. It's there in my afterward, right? I kind of say that, yes, I've used these two viruses very specifically. One of the things that came up to me is what a negative role prejudice of all different sorts, prejudice towards ideas, the notion that a virus was infectious, so prejudice against this one idea sort of put a stumbling block, not just against the cancer virus story, but it repeats itself with AIDS, HIV/AIDS, where, a scientist kind of said poppers are what are causing this, not jalapeno poppers, but anyway, and not Karl Popper either. But poppers were what were causing this, and it couldn't possibly be a virus. So there was that prejudice, but there was also, of course, within the US anyway, the prejudice against male homosexuality. So the research wouldn't progress in a certain way. So that's kind of thing. But to get back to how courses and to teaching about, so viruses, things continually are being caused by viruses and people don't believe it. And so one has to keep reconsidering what a virus is. And I think it goes down to eventually people's misconceptions about what is a virus compared to what is a different kind of pathogen. And I think that's something even a lot of doctors, even a lot of researchers who work with viruses sometimes forget. I was chatting with somebody the other day who, when I asked him what was a virus, he started to name bacteria as examples, then I and he goes, oh my goodness, and then was mighty embarrassed. So, and then backtracked, but, and then of course corrected himself, or herself. But so understanding what a virus is and the implications for disease, I think is one big takeaway from my book that I think people should walk away with. In terms of using APS resources, so this, as you mentioned correctly, is a pet project, right? How can one, I think that resources like archives are sometimes left too late in an education in history, especially history of science. We aren't taught as undergrads typically to use archives. We're told about them. Well, I didn't study history of science as an undergrad, but I don't think enough of us make enough use. And when one is in Philly with such a embarrassment of riches of archives, I think one should just have classes that are based in archive. Let the students loose on, granted, you curate what they're able to access. And of course, you also have, one has to curate, be careful about what they're allowed to touch and feel and how much the archive hours, but with the right kind of permission, students should be able to look through and learn the process of, okay, here's an archive, here are the papers, let's look through X amount of stuff with certain amount of preparation and primary, reading of primary source materials, but then start to look and define a problem. Here's an interesting resource. I mean, I wrote a whole paper in less than a year. It was one of the fastest turnarounds in my history, for sure, of, working December and January of 2014-15, and by December 2015, the paper had been submitted. By January, it had been accepted, right, in 2016. So it was that quick of a turnaround based on that one letter, that Christmas fairy story. So you could, I mean, undergrads have wonderful opportunities. One could show them something you worked with, but I think even more exciting is to let them have at an archive that is still being put together so that they find things and what is this? And then you bring it to discussion in the classroom. I mean, the thing to do, of course, is to find something and then discuss it as a class and then write something about it. Because for me, and I'll bring in the other element of teaching, for me, I think it's fairly evident from the book. I'm always thinking of things as a writer, as well as a historian. So telling a story, how does one write a story? And I think for students to think that way with an archive, what's the story? And to build a story out of a source is a good kind of heuristic or is a good way to think. And it's a good way to teach also, for them to, for us as teachers to tell them to think in terms of stories can really help them focus. And archives are great sources for stories.
And I think that brings us back to Dickens. And thinking about the role of narrative, both in literature, but also in the way that we tell histories, and finding those little nuggets, those little bits that can get cited and requoted. I think it's at the heart of the story that you're telling. It's been a pleasure chatting with you. It's always lovely to see you. I encourage everyone to pick up a copy of Neeraja's book and look.
There's a discount.
There is a discount, so that information is in the chat. Thanks again for joining us. Our next virtual discussion will be on October 6th, and information about the event is on our website. But for now, Neeraja, I'll let you get to bed because I think it's very late where you are. So
no, it's only 8 o'clock.
Okay, fine. Lots of time. Anyway, thank you again.
Okay, bye-bye.
Take care.